Active transport is the process by which dissolved molecules move across a cell membrane from a lower to a higher concentration. In active transport, particles move against the concentration gradient and therefore require an input of energy from the cell. There are two types of active transport. First is bulk transport and second is ion pump. Let's discuss about bulk transport first. Movement of large ions, molecules, or particles dissolved or carried in a medium is called bulk flow. Rate is determined by the differences in hydrostatic or air pressure. We have two types of bulk transport called endocytosis and exocytosis. First, we have endocytosis. Endo, which means internal, and cytosis, which means transport mechanism. Endocytosis is a general term for the various types of active transport that move particles into a cell by enclosing them in a vesicle made out of plasma membrane. The second type of bulk transport is exocytosis. Cells must take in certain molecules, such as nutrients, but they also need to release other molecules, such as signaling proteins and waste products, to the outside environment. Exocytosis, exo, which means external, and cytosis, which means transport mechanism, is a form of bulk transport in which materials are transported from the inside to the outside of the cell in membrane-bound vesicles that fuse with the plasma membrane. Kinds of endocytosis. First, we have phagocytosis. Phagocytosis, which means cell-eating, is a form of endocytosis in which large particles, such as cells or cellular debris, are transported into the cell. This is the type of endocytosis used by the macrophage to engulf a pathogen.
Once the cell has successfully engulfed a target particle, the pocket containing the particle will pinch off from the membrane, forming a membrane-bound compartment called a fluid vacuole. The fluid vacuole will later fuse with an organelle called a lysosome, the recycling center of the cell.
Second is pinocytosis. Pinocytosis, which means cell drinking, is a form of endocytosis in which a cell takes in small amounts of extracellular fluid. Pinocytosis occurs in many cell types and takes place continuously, with the cell sampling and resampling the surrounding fluid to get whatever nutrients and other molecules happen to be present. Pinocytous material is held in small vesicles, much smaller than the large fluid vacuole produced by phagocytosis.
Last but not least is the receptor-mediated endocytosis. Receptor-mediated endocytosis is a form of endocytosis in which receptor proteins on a cell surface are used to capture a specific target molecule. The receptors, which are transmembrane proteins, cluster in regions of the plasma membrane known as coated pits. This name comes from a layer of proteins called coat proteins that are found on the cytoplasmic side of the pit. When the receptors bind to a specific target molecule, endocytosis is triggered, and the receptors and their attached molecules are taken into the cell in a vesicle. The coat proteins participate in this process by giving the vesicle its rounded shape and helping it bud up from the membrane. Receptor-mediated endocytosis allows cells to take up large amounts of molecules that are relatively rare in the extracellular fluid. Although receptor-mediated endocytosis is intended to bring useful substances into the cell, other less friendly particles may gain entry by the same route. Flu viruses, diphtheria, and cholera toxin all use receptor-mediated endocytosis pathways to gain entry into cells.
Is the ion pump. The mechanism of ion pumps is generally complicated. It is useful to think of the enzyme as being characterized by a limited number of conformations to which we can give identifying labels. The reaction mechanism is then viewed as the sequential steps that occur among these conformations to achieve ATP hydrolysis and ion transport. Transformations between these conformations are determined by rate constants. Each step has two rate constants, one for the forward and the other for the reverse reaction.